work on DNA damage responses in the mammalian cells, and then today's title is RTEL1 helicase regulates homologous recombination in the mammalian cells during DNA replication. So the genetic material which is stored in the form of DNA and chromosomes in all our cells is susceptible to various types of damage. And among different types of DNA damage, DNA devastating breaks are the most dangerous types of lesions. So which can arise by endogenously by free radicals and then other starred or collapsed application force mechanisms. And even exogenously by radiations or chemicals, it can induce DNA devastating breaks. The first thing that cell responds to DNA devastating breaks is activation of the cell specific checkpoint, which actually arrests the cells in the at a particular stage, either in the G1 or in the S phase, and then accordingly facilitate the repair by the repair pathways. There is very good coordination between cell cycle checkpoints as well as repair pathways in order to take care of repairing these breaks and then maintaining the genome integrity. If there are unrepaired breaks or misrepaired breaks, basically it can lead to genome instability, which is the hallmark of the cancer cells and will induce tumorigenesis. There are two major pathways to take care of repair of the DNA devastating breaks. One is the DNA non-homologous enjoining, which is generally often error prone because of the microinsertions or deletions basically at the break points. So as opposed to NHJ pathway, there is another classical homologous recombination pathway, which always requires homologous template DNA to repair the breaks. And then, so this is considered as an error-free pathway in the repair of the DNA devastating breaks. It always requires the processing of the DNA ends to generate a 3' single-strand DNA, to which the recce protein in the bacterial system and its functional ortholog in the family system is the RAD51 protein, which actually polymerizes onto the single-strand DNA, which actually searches for the homology, invades the homologous duplex DNA to generate a D-loop structure, and this recombination intermediate is resolved in two alternative pathways to complete the repair in a error-free fashion. Many genes are known to participate in the homologous recombination mediated repair of the DSPs. So mutations in some of these genes are known to cause various genetic diseases as well as cancer. Some are examples are here. Basically, the BRCA1 as well as BRCA2 general mutations leads to breast as well as ovarian cancer, predominantly women. And there are 22 FANG genes basically. Any one FANG gene mutation leads to Fanconi anemia, and helicase mutations like the BLM gene and then Werner gene leads to Bloom syndrome, Werner syndrome. And protein kinase mutations leads to ATM mutation leads to ataxia telangiectasia, and then ATR kinase mutation leads to ATR sickle syndrome. And mRNA complex protein NBS1 gene mutation leads to Nidme genetic syndrome. These are all chromosome instability and then cancer susceptibility genetic diseases. So that shows the importance of the homologous recombination in taking care of the repair of the DSPs as well as in maintaining the genome maintenance. The RAD51 is the key player, which is the central key player in the homologous recombination mediated repair of the DSPs, which actually polymerizes onto the single-strand DNA to form a nucleoprotein filament, so which actually searches for the homology, invades the homologous duplex DNA, and then initiates the homologous recombination. But the RAD51 filament formation is actually rather inefficient in the cell because there is a RPA protein, which is a single-strand DNA binding protein, which actually binds to the single-strand DNA with a higher affinity, and is also present abundantly in the cell basically. To overcome this barrier from the RPA protein, there are recombination mediators. Those are like the BRCA1, BRCA2, as well as RAD51 paralogs basically. In mammalian cells, there are 5 RAD51 paralogs. These are named as RAD51B, RAD51C, RAD51D, XRCC2, and then XRCC3. If you look at the protein sequence, it doesn't show high degree of conservation to the RAD51 except for the two motifs which are shown in the red boxes. These are Walker A as well as Walker B motifs, which are required for the ATP binding as well as ATP hydrolysis. If we make a mouse knockout of any one of the RAD51 paralogs, that actually leads to embryonic lethality, indicating their essential genes. But the bigger question is that the essential functions in the genome maintenance is largely still unclear. And any one gene mutations leads to different types of DNA adaptive sensitivity in the context of different chemical agents basically, and it shows spontaneous chromosomal aberrations in the form of chromatid breaks, radials, and then gaps basically, and then reduction in the homologous recombination. So, and germinal mutations in the paralogs are also known to now in the recent times, cause basically breast as well as ovarian cancer in the, in the females basically. And two of the paralog mutations, that is RAD51C as well as XRCC2, so leads to Fanconi anemia. And biochemical studies from a couple of groups basically shows that these paralogs exist majorly in two complexes. One is the BCDX2 and other is the CX3 complex. So in our lab, so we showed a so a new functions basically, these are repair independent functions in taking care of the replication problems in the genome. So what happens is that if there is a DNA lesion basically, so the folks actually undergo remodeling by the helicases as well as even, the RAF51 as well as RAF10 paralogs. And these RAF51 as well as RAF10 paralogs actually, so, binds to these reversed forks basically and stabilizes them from degradation by the nucleases. That's how it actually maintains the integrity of the genome. So in addition to taking care of the stabilization of the stalled forks, these paralogs are also involved in the restart of the stalled replication forks. So by homologous recombination pathway, and even if there is a lesion and then that gets converted to replication association space, so that also is taken care in a recombination mediated replication restart, and then accordingly, complete the genome duplication and then maintain the integrity of the genome. So all the homologous recombination, and then so the factors which are involved in the homologous recombination is actually important for taking care of the repair of the DSPs as well as the replication problems in the genome. Basically, their activity has to be controlled, regulated. Otherwise, it can lead to various types of genome rearrangements. Basically, like the gene amplification, loss of genetic information, loss of heterozygosity, and then different types of chromosomal rearrangements. So there has to be a proper balance between the, homologous recombination pathway and then the homologous recombination proteins like the RAF1, RAF2 as well as RAF10 paralogs, and along with the anti-recombinases. These are all the helicases like the RTEL1, RE, FPH1, BLM, RECUL5. They're all helicases basically, which are, their functions has been implicated as anti-recombinases basically in trying to regulate the homologous recombination in our cells. So, two papers basically from the Simon Bolton group showed that RTEL1 helicase actually is important for suppressing the homologous recombination during DSP repair. So, subsequently, they also showed that RTEL1 helicase is a PCN interacting helicase, which is actually required for the genome wide replication and then taking care of, genome duplication as well as genome maintenance, basically. So, we speculated that possibly RTEL1 helicase may be regulating the homologous recombination during DNA replication. So there are multiple factors which are actually required for the DNA replication in the mammalian cells. So the most important ones are the MCM helicases, and then the Pol epsilon, which is required for the leading strand synthesis, Pol delta for the lagging strand DNA synthesis, PCN is the clamp basically, and you have the RPA protein. So, this replication is actually not a very, a smooth process basically in our cells because, it actually goes through, so with the template damage in the DNA, so if there is a DNTP alterations in the cells that actually leads to replication stress, if there is a secondary structures like the G4 structures, which actually forms a roadblock for the DNA replication, there's a transcription, replication, collisions basically, all this actually leads to replication stress in the genome. So that is where the homologous recombination proteins actually come into the picture in taking care of dealing with the replication problems in the cells, majorly in taking care of stabilization of the stalled DNA replication forks and as well as in resuming the DNA replication. We speculated that possibly the RTEL1 helicase may be regulating the homologous recombination during DNA replication. To study this basically, Kumar, who started this work, so we carried out I-bond experiment, that is isolation of proteins on the nascent DNA. So what you need to do is that label the replicating sites with the IDU, which is a halogenated thymidine analog, and then treat it with the hydroxyurea, which actually causes genome-wide replication stress, and then later do the pull down with antibody specific to the IDU, and then later, do the Western blotting with all these known factors basically. You can see that RTEL1 is actually enriched in the recovery forks basically. In addition to RTEL1, interestingly, we also see accumulations of the HR factors like the RAF1 and even the RAF1 paralogs. Later, to understand the role of the RTEL1 helicase in the replication context, basically, we subjected the cells to hydroxyurea as well as ofidicolin. Both actually causes replication stress. And when we measure the cell survival, basically, upon depletion of the RTEL, there is a high degree of cell death. But we don't see the cell survival sensitivity in the context of the zeosine, which actually induces DSPs, this actually gives us the separation of some functions basically, its role in the replication stress context, but not in the DSP repair context. So then subsequently, Suruchi carried out further experiments basically. We measured the global DNA replication by the BRD incorporation. compared to the control cells, as you can see that the flow cytometry studies basically, here's the quantification, you can clearly see that there is about 50-fold reduction in the global DNA replication compared to the control upon depletion of the RTEL1 with the two shRNAs. That shows that RTEL1 is important for DNA replication in the human genome. So because RTEL was already implicated as a anti-recombinase, we wanted to measure the homologous recombination. For that, we used a reporter system wherein this contains two copies of the gene coding for the green fluorescent protein. Both is actually mutated. First one has a 5' truncation, and second one is full length actually, but is interrupted with IISc1 recognition endonuclease. So this reporter has been integrated in the human chromosomes, basically in the U2S cells. And then, so we measured after inducing the break with ISC1 transfection with a plasmid encoding ISC1, so endonuclease, which actually makes a break here. And if the repair occurs by the homologous recombination pathway, then you see the generation of the GFP positive cells, which can be measured by the flow cytometry. And here is the data. You can clearly see that compared to the control cells, upon depletion of the RTEL1, you can clearly see that there is 4 to 5-fold increase in the homologous recombination. Now, that can be rescued upon co-depletions of the RAFT1 paralogs like the RAFT1C, XRCC2, and then XRCC3. So this is in the context of the DSP repair. Now we wanted to study homologous recombination in the context of the whole genome during DNA replication basically. So what we did is that we subjected the cells to hydroxyurea, which actually causes genome-wide replication stress. And then in that context, basically using the same reporter, we scored for the GFP positive cells. So you can clearly see that compared to the control cells, RTEL1 depleted cells shows 4 to 5-fold increase in the homologous recombination, which can be rescued upon co-depletion of the RAFT1 paralogs. This clearly shows that in the absence of the RTEL1 helicase, there is high degree of homologous recombination events basically, which can be rescued upon co-depletion of the HR factors like the RAFT1 paralogs. So in the context of increasing the homologous recombination, we also see increasing the RAFT1 foci formation compared to the control cells. Basically, in the RTEL1 depleted cells, you see increasing the RAFT1 foci formation. That again we can try to rescue upon co-depletions of the RAFT1 paralogs. Now this clearly tells us basically there is hyper recombination events are happening in the whole genome basically when there is no RTEL1 helicase. So if that is the case, can we rescue the replication defect in the RTEL1 depleted cells by co-depletions of the RAFT1 paralogs? That's exactly what we tested basically. So we performed DNA fiber studies. Again, what we do is that we label the replicating sites with the two halogenated thymidine analogs, that is the CLDU as well as IDU. And then, so the replication can be studied at the single molecule level. And when we score for about DNA, about 200 DNA fibers basically, here is the raw data. You can clearly see that compared to the control cell, the global DNA replication is actually reduced upon depletion of the RTEL1. Now, this replication defect can be rescued upon co-depletion of the RAFT1 paralogs, including RAFT1 co-depletion. That clearly suggests that basically there is hyper-recombination events are happening, which is actually now interfering with the DNA replication in the absence of the RTEL1 helicase. In fact, the the sister-to-fork ratio basically, which is again increased upon depletion of the RTEL1 helicase, that sister-to-fork ratio also can be rescued upon co-depletions of the HR factors like the RAFT1 paralogs as well as even RAFT1. What is actual, so the direct evidence basically in the absence of the RTEL1 helicase, there is hyperrecombination events are happening basically. Whether these paralogs or even the RAFT1 protein is actually persistent at the sites of the replicating sites. So to address this basically, we performed iPond experiments. This is isolation of proteins at the replicating sites. What we do is that again, we label the replicating sites with the halogenated thymidine analogs. This is just a DNTP analog. And then, so we do the pull down with the ideal specific antibody. And then, so we plotted for various proteins basically. So like the RAFT1 and the RAFT1 paralogs. Compared to the control cells, if you notice in the RTEL1 depleted cells, you see that compared to the control, so RAFT1, the levels are actually increased in the absence of the RTEL1. Similar to RAFT1, we also see the enrichment of the RAFT1 paralogs like the XRCC2, XRCC3 as well as even RAFT2 protein. So this is the direct evidence that in the absence of the RTEL1 helicase, the HR factors actually persistent at the replicating sites and which is actually interfering with the DNA replication. As a result, now when we co-deplete the HR factors basically, now you can rescue the DNA replication defect in the RTEL1 depleted background cells. So RTEL1 is a PCNA interacting helicase basically. So we wanted to test whether its interaction is required for suppressing the homologous recombination during DNA replication. So we made a isoleucine to alanine. So in the PCNA interacting motif basically. So we studied this. So the HR events using the reporter system. Here is the GFP positive cells. Compared to the control cell, there is 4 to 5-fold increase in the GFP positive cells. That means HR events are high basically, which can be rescued upon wild type expression of the RTEL1, but not the PCN interacting motif basically. So we also wanted to understand the helicase function of the RTEL1 in suppressing the homologous recombination during DNA replication. For that, we made two mutants basically, that is lysine to alanine as well as lysine to arginine in the Walker A motif basically, which is actually required for the ATP binding as well as ATP hydrolysis. Again, these Walker motif mutants are actually defective in suppressing the hyper HR, so the cells basically. So now, using these mutants, we wanted to understand, so the DNA replication basically, yes, replication defect is there upon depletion of the RTEL1, which can be rescued upon expression of the wild type RTEL1, but not this PCN interacting motif mutant or even the Walker motif mutants. That suggests that RTEL1 interaction with PCNA as well as its helicase activity is actually important in suppressing the homologous recombination during DNA replication and taking care of genome duplication. So RTEL1 genetic mutations leads to to two disorders called the higher hydroxyl syndrome. Sorry. So as well as dyskeratosis congenita, these are developmental disorders with various clinical features, including premature aging. So and then bone marrow failure, and patients are also susceptible to developing glioblastoma. So far, about 25 mutations have been identified in these patients basically, and which is actually spread across whole entire helicases basically. So these are all point mutations, missense mutations basically. Now we are trying to understand the functional defect associated with these mutants, and trying to understand how they function in the context of suppressing the HR as well as in taking care of the replication problem. So just to conclude that RTEL1 helicase is required for the normal DNA replication in our cells, and then hyperhomologous recombination as well as replication defect in the RTEL1 depleted cells can be rescued with a co-depletion of the RAT1 as well as RAT1 paralogs, and then RTEL1 PCNA interaction and then helicase activity is required for suppressing the homologous recombination during DNA replication. So this work we have communicated recently. And this is my current team. And Kumar started the work, and then Suruchi later, carried out majority of the work what I presented. In addition to Suruchi, Satyaranjan as well as Tarun also contributed to this work. And then I greatly applaud many scientists outside India as well as within India for providing reagents for our studies basically. And I greatly applaud the funding support from various sources. And thank you all for your attention, and then happy to take question.
Is one phenomena called chromothripsis. Yeah. Right. So in many cancers, that is application. That must have to do something with this protein. Do you have any comment on that?
It's a interesting thing. Absolutely. So it is the complete shattering of the chromosomes basically. So we do see increase in the chromosomal aberrations, but specifically, so we are not actually addressed. I don't think there is actually chromothripsis happening because cells are able to survive with a compromised DNA replication. So, and with RTEL single depletion, we do see chromosomal increase the aberrations basically. That aberrations actually further increases upon co-depletions of the HR factor. That shows that you need to have the fine balance between HR proteins as well as anti-recombinases. So, in the absence of both, basically, you see further increase in the genome instability, but at the compromising, compromise of the cell survival with an instability, which could trigger tumorigenesis.
I'm not a biologist, so sorry if it is a live question. You have said that you have implicated RTEL1 helicase in this. What would be the mechanism? What does it actually do?
That's a very good question, actually. So one study by Simon Bolton group, they show that it actually disrupts the D loop, basically. There are other helicase mechanisms wherein what it does is that it actually strips the rafting from the single-stranded DNA. But that kind of an activity is so for the RTEL1 helicase, people are not sure. So that is what is some kind of an implication that what we have basically. But it is known to disrupt the D loop basically. Whether it has a role in disrupting the RAF1 filament from the single strand DNA is something to be studied.
So it's the helicase which is unwinding vesicle and
Yes. Exactly. Because when we make a helicase mutants, it is disfined. It is actually doesn't do its function. Basically.
Professor, okay.
Very nice results and presentation. Couple of general questions. Is RTL found across both prokaryotic and eukaryotic?
Not in the prokaryote, but in the higher order organisms. Only the higher.
No sign of it in prokaryotes.
No, this is RTEL is basically the name goes with a regulator of telomere elongation. So it is generally present in the mammalian cells.
Second question was, does it have any influence on non-homologous enzyme?
RTEL1 not for our knowledge basically. It's only in the HR context.
And what exactly is the helicase activity doing? Was it promoting expression of HR specific proteins?
No, it is actually not involved in the expression of the HR factors basically, but it regulates the HR factors by possibly. So as he was asking, what is the mechanism? Maybe it actually unwinds the D loop. But in addition to that, it might have a role in stripping of the RAF1 and then RAF2 paralogs from the single strand DNA at the replicating sites. If that is not controlled basically, then you see persistent HR factors. So doing its hyper recombination events, which is actually possibly the reason for reduction in the global DNA replication.
Professor Majumdar.
Just a quick question. If you have mutant RTEL1, you're going to suppress replication and you're going to also suppress homologous recombination. Is the suppression of homologous recombination a consequence of reduction in replication?
Because of the hyper HR, replication is actually compromised basically. That is because of the hyper recombination events, because there is a persistent HR factors at the replicating sites. That is in the absence of the RTEL1. So now that replication defect you can rescue by co-depleting the HR factors. That's how we are able to see.
Yeah, I know, but what I'm saying is that is the suppression of the homologous recombination a consequence of suppression of replication?
Suppression of homologous recombination. So we see increase in the HR. So in the absence of RTEL, which you can rescue with HR factors basically. So that because of the increase in the HR, there is a replication defect basically. That replication defect you can rescue basically. But what happens is that's what Samit was also asking basically. The chromosome instability basically increases when you don't have both the proteins. With singly also, yes, you see increase in the genome instability. And if you have a co-depletions basically, chromosomal aberrations actually further increases. But with replication is rescued, but with an accumulated, so instability and then mutations basically, which possibly can lead to, cancer.
So the paralogs, are they redundant in their function?
That's an excellent question. Any single knockout is basically embryonically lethal. Although many functions it shows sort of similar function, but one cannot compromise with the another one. So in the larger context, out of 5 paralogs, there is a BC subcomplex, DX2 subcomplex, and CX3 subcomplex. If you don't have one, the other partner actually undergoes destabilization. The protein levels actually reduces. So it functions in a complex, and then that is possibly some redundancy is there, but so it cannot compensate for one mutation with another paralog.
You seem to have mentioned the use of hydroxyurea for genotoxic stress. How serious is that? Why I ask is because hydroxyurea is a pretty common management tool for sickle cell disease.
Yeah, you're correct. So with sickle cell anemia, so I don't know at least mechanistically how it will actually, it helps in for the treatment.
As far as it's known, it increases, enhances the expression of fetal hemoglobin.
But as such, it's an inhibitor for the ribonucleotide reductase, which is the enzyme which is required for the DNTP synthesis. When the DNTP synthesis is actually reduced, then naturally you will have the replication problem in the gene. That's how we use as a replication poisoning agent.